Hello, my name is Olga Zamudio, and today I'm going to give you a tour around the protein world and Japanese culture. Japanese have a special word to describe the beauty behind the ephemeral, Wabi-Sabi. It is about the aesthetic of things in existence that are imperfect, impermanent, and incomplete. Origami is a good example of this, and so is the lifespan of a protein in your body. Today, I'm going to talk about the ephemeral life of a protein, origami, and when things derail. Maybe the first thing that pops into your head when you hear the word protein is food related: eggs, cheese, meat. But proteins have a plethora of roles in your body, from making your bones strong to identify and attack pathogens, or to break down the sushi you had for dinner. All proteins the same place, as basic instructions in your DNA. When the time comes, those instructions are copied and sent to the protein factory, the ribosome. The ribosome will make long chains of amino acids, the sheets of paper for our origami analogy. But a sheet of paper per se is useless. What happens next is what matters: folding. You see, the function of a protein is in their structure. Only when it's correctly folded it will perform their function properly. Some proteins fold as soon as they leave the ribosome. They are expert yogis and need no assistance, from paper square to crane in nanoseconds. Some others need a little hand-holding. A special kind of protein, appropriately called chaperones, will accompany and look after them until they are ready. Chaperones will attach to the immature protein and prevent improper behavior, because when these proteins are out of the protein factory, they are very sticky. Without chaperones, they can create a big mess. Maybe this relates more to an analogy with high schoolers than origami, but the general idea is that chaperones will prevent our protein to be crumpled. Finally, some proteins will need the direct intervention of an origami master. Once they leave the ribosome, they may fold partially, and then this huge, massive, for protein standards that is, barrel-shaped protein called chaperonin comes to engulf the semi-folded protein, puts a lid on it, and releases it from the bottom, perfectly folded. Magic! The journey of a newly folded protein is just the beginning. They will be carried away by other proteins to the different compartments of the cell, where they will perform their job, whether it is to be a building block, a karate chopper, a gatekeeper, a snitch, a construction worker, a chaperone, or an origami master. In mammals, the lifespan of a protein can go from 0.5 to 35 hours in dividing cells and around 43 hours in non-dividing cells. When a protein gets older, a snitch informs the cleaning mob to get rid of them. The cleaning mob, aka the ubiquitin proteasome system, or UPS, will chop down problematic, inefficient, or defective proteins into amino acids to be reused. Believe me, it is not pretty and can be gruesome. Then the cycle starts over and over again. From time to time, a rebel protein will behave inappropriately, either by folding incorrectly or not folding at all. Sometimes chaperones and chaperonines can solve the problem. When they can't, riot. The harmful effect of misfolding may be due to loss of function or formation of aggregates. A loss of function occurs when the instructions miss a step or during the process a mishap happens. You may end up with a bird instead of a fish. It won't work the same. On the other hand, an unfolded or partially folded protein is like working with a tissue paper and white glue. It's messy. Soon proteins will start to stick to each other, creating lumps or plaques and getting stuck in places. Not even the proteasome can get rid of them easily, so these aggregates start to accumulate. For us, humans, this translates into disease. Susan Linsquith, an American scientist, said that protein misfolding could be involved in up to a half of all human diseases. The list includes known foes: Alzheimer's, Creutzfeldt-Jacobs, Parkinson's, Huntington's, diabetes, cystic fibrosis, sickle cell anemia, and even some types of cancer, all related to misfolded proteins. Take Alzheimer's disease, a progressive degenerative disease that damages neurons, leading to a memory impairment and sometimes dementia. In 1906, Dr. Alois Alzheimer examined the brain of a dead patient suffering a strange mental illness. Her brain showed weird clumps and entangled fibers made of aggregated protein. In Alzheimer, the connection between neurons is also lost, impeding the brain to work properly. Alzheimer starts with the accumulation of misfolded beta amyloid protein, triggering inflammation and cell death. Indeed, the presence of misfolded protein in blood is a high-risk marker for developing Alzheimer in the future. Recent efforts focus on the use of chaperones to improve beta-amyloid transport to degradation compartments in the cell. Because the Japanese are right, Wabi-Sabi, there is beauty in the temporary, and proteins shouldn't stick for too long. They should be like origami, ephemeral, that can be destroyed by the thump of a fist. And so should be protein origami, fugacious and gorgeous. Thank you.